A microscopic killer has been lying in Siberian permafrost since before modern humans built a single city, planted a single crop, or recorded a single word of history. And global warming, the thing you'd expect to wake it up almost, isn't the reason it's stirring. Something far stranger already did the job, and it's not melting ice. It's us. Most people imagine a simple story: the planet heats up, the polar ice melts, and a forgotten plague drifts out to sweep across the globe. That story is mostly wrong. The real mechanism and what these things actually look like under a microscope is stranger, slower, and far more disturbing than the disaster movie version. In our videos on this channel, we usually measure time in centuries and empires. But beneath the Siberian tundra sits a vault where time simply stopped, some of it over 48,000 years ago, and its contents are starting to move. For most of modern biology, viruses had a reputation: tiny, primitive, barely alive. A single strand of genetic code so small you need an electron microscope just to see its blurry outline. A standard human flu virus, for instance, carries a genome of only about 8 genes. It's less a creature than a whisper. That rule started cracking in the early 1990s, though nobody realized it yet. Scientists sampling a water cooling tower in Bradford, England, found something living inside an amoeba that looked, under the microscope, exactly like a small bacterium. It sat there, unidentified, for over a decade because nobody expected a virus to be that big. It wasn't until 2003 that researchers finally proved it wasn't a bacterium at all. It was a virus, just 1-3 times larger than anything previously known, with a genome carrying over 1,000 genes. They named it mimivirus, short for microbemimicking virus, and it became the first confirmed member of a category nobody had taken seriously: giant viruses. 5 years later, the same French research group found something even stranger. While studying a related giant virus, dubbed mammavirus, pulled from a Paris cooling tower, they noticed something living inside it: a second, much smaller virus, hijacking the giant virus' own reproductive machinery to make copies of itself. They called it Sputnik. It was the first confirmed case of a virus infecting another virus, and it opened up an entirely new branch of biology that, until that point, nobody knew existed. So by the mid-2000s, virologists already knew giant viruses were real and knew they had parasites of their own. What they didn't know yet was how far back in time these things could survive. That question sent them looking in a very different kind of place: not a cooling tower, but underground, in Siberia, in ice that hadn't seen daylight since the last ice age. Before the permafrost search even began, the same team made one more discovery that reframed the whole picture. In 2013, working thousands of miles apart, they pulled 2 more giant viruses out of ordinary, present-day sediment, one from a river mouth on the coast of Chile, the other from the mud of a freshwater pond outside Melbourne, Australia. They called this new family Pandora virus for its container-like shape and its equally strange genetic content. Some individual species carry up to 2,500 genes, more coded instruction than several free-living bacteria manage with. Genetically, over 90% of a Pandora virus genome matched nothing else in any database on Earth. That mattered because it proved giant viruses weren't some Siberian anomaly, a fluke of deep freeze and isolation. They were already living in warm mud on 2 different continents, in plain sight, the whole time. The Arctic didn't invent them. It just happened to be extraordinarily good at keeping them exactly as they were. In 2014, a team led by Dr. Jean-Michel Claverie at Aix-Marseille University drilled into Siberia's ice-rich permafrost, known as Yedoma, roughly 100 feet below the surface. What they pulled out was a specimen that had been sealed in total darkness for around 30,000 years later, named Pithovirus sibericum. Under a standard light microscope, Pithovirus doesn't look like a virus at all. It looks like a dark, oversized, jar-shaped vessel almost identical to an ancient Greek amphora urn with a cork-like plug sealing one end. Its genome carries roughly 500 genes, 60 times more genetic instruction than the flu virus most of us catch every winter. It wasn't even the most complex thing they'd found. A related giant, Pandora virus, discovered by the same team the year before, carries up to 20,500 genes, more coded instructions than some free-living bacteria. 2 very different giants, both waiting in the same frozen ground. That raised an uncomfortable question after 30,000 years in the dark: Was any of it still alive? There was only one way to find out, and it came with a lethal design problem. How do you test whether a revived, 30,000-year-old organism is still infectious without risking exposure to something nobody has an immune defense against? Clavery's team solved it with a decoy. Instead of exposing human or animal cells, they offered the specimen a single-celled organism called acanthamoeba, a harmless amoeba that exists purely to test whether ancient material can still infect anything at all. It became, in effect, the ultimate biological tripwire. What happened next was fast. As the ice thawed and the virus rehydrated, it didn't stumble back to life, it moved with precision. The amphora-shaped shell aligned with the amoeba. The cork-like plug popped open. The viral wall fused directly to the host's outer membrane, and in a single motion, the genetic core was injected inside. From there, it hijacked the amoeba's internal machinery, halting its normal function and forcing it to mass-produce copies of the virus. Within about 12 hours, the amoeba was so packed with new viral particles that its membrane ruptured, releasing a fresh swarm into the surrounding water. An organism that had been dormant since before cave paintings were still wet woke up in a modern lab, found its target, and executed its biological code exactly as it had 30,000 years earlier. Cold, dark, oxygen-free permafrost hadn't just preserved it, it had paused it. Pithovirus wasn't alone in that Siberian sample either. The following year, in the same permafrost layer, Clavery's team isolated a completely different specimen, a spherical giant they named Mollivirus sibericum. Different shape, different genetic architecture, same 30,000-year-old ground. Two unrelated giant viruses buried side by side, both still capable of infecting a living cell the moment the ice let go of them. Whatever was preserving them wasn't preserving one lucky survivor. It was preserving an entire indifferent cross-section of ancient life. That 30,000-year figure didn't hold for long. In 2023, Clavery's team returned to Siberia and pulled samples from 7 different sites, remote lake sediments, mammoth remains, riverbank permafrost, and isolated 13 more viral strains, representing 5 virus families that had never been described before. The youngest, recovered from the stomach contents and coat of a preserved woolly mammoth, was about 27,000 years old. The oldest came from sediment 16 meters, down inside an ancient underground lake, nearly 48,500 years old, and a member of the very same Pandora virus family first found in warm coastal mud a decade earlier. The oldest confirmed infectious virus on the planet, and it belonged to a genus that also lives comfortably in a pond outside Melbourne. Every one of those 13 strains, once thawed and introduced to amoeba cultures, infected and killed their host. None of them had evolved, mutated, or degraded in any meaningful way in tens of thousands of years. They had simply waited some for over 48 millennia for something to reach them. While Clavery's giants stayed contained behind laboratory glass in Marseille, the summer of 2016 proved the vault in Siberia was already leaking into the world outside it. On the remote Yamal Peninsula, an unprecedented heatwave baked the tundra far above its historical average. As the topsoil softened, it exposed the frozen carcass of a reindeer that had died 75 years earlier, and with it, dormant spores of Bacillus anthracis, the bacterium behind anthrax. The spores reactivated. Within days, the outbreak swept through the region. More than 2,000 reindeer died. Dozens of nomadic herders were hospitalized with fever and lesions. A 12-year-old boy lost his life. The Russian military deployed specialized biodefense units to incinerate carcasses, vaccinate surviving herds, and evacuate entire communities. Headlines worldwide ran with a simple, terrifying narrative: melting ice caps were about to release forgotten plague smallpox relatives, extinct flu strains, Paleolithic killers onto a world with no defense against them. It's a great headline. It's also not quite what's actually happening. A quick pause here, because this is the point where most videos on this subject go off the rails. Here is the part the panic headlines skip. Surface warming, no matter how brutal the summer, can only thaw the active topsoil, a thin layer just a few feet deep. The oldest, most dangerous material, the 30,000 TO 48,500-year-old giants, and whatever else sits beside them, is locked dozens of meters below that, in permafrost so cold and so deep It would take centuries of steady warming to reach it naturally. Climate change alone isn't digging fast enough to open that vault. Something else is already doing it on a much shorter timeline. Siberia isn't just an expanse of empty snow. It's one of the most resource-rich regions on Earth, vast reserves of gold, petroleum, natural gas, diamonds, and the rare earth minerals that modern technology depends on. As surface ice recedes, mining and energy companies are moving into the Arctic with industrial equipment at a scale the region has never seen. To reach those deposits, open-pit operations don't wait for a heat wave. They strip the topsoil and cut trenches dozens of meters deep into Pleistocene-era ground soil that hasn't seen sunlight since Neanderthals were still alive. Every day, excavators churn up thousands of tons of material that used to be a sealed vault. When that soil ends up in open-air runoff ponds exposed to summer heat, whatever was frozen inside it washes straight into local river systems. We're not just extracting minerals. We're physically unearthing forgotten eras of biology on an industrial schedule, not a geological one. Climate change might take 300 years to reach a layer of ground. An excavator can reach it in an afternoon. It gets stranger. Giant viruses are so large and genetically complex that they aren't even at the top of their own food chain. Remember Sputnik, the virus that infects a virus discovered back in 2008? It turned out not to be a one-off curiosity. Researchers have since found that many giant viruses carry their own dedicated parasites, a whole category now known as virophages. A virophage can't infect a normal cell on its own. It's too simple, too stripped down, missing the machinery to do it alone. Instead, it waits for a giant virus like mamavirus or pithavirus to invade a host cell first, then quietly hijacks the giant virus' own reproductive factory to make copies of itself, weakening its host and cutting its infectious output by as much as 70% in the process. Buried in that permafrost isn't a random scatter of particles. It's an entire layered microbial ecosystem predator, prey, and parasite that has been sitting in the dark for tens of thousands of years, fully intact, waiting for the sound of drills. It's worth pausing on the people who study this for a living, because their take is more measured than the headlines. Eugene Koonin, an evolutionary biologist at the U.S. National Institutes of Health, who has studied giant viruses for years, has been blunt about it. Finding these things is not going to cause a widespread epidemic. And Eric Delwart, a molecular virologist at UC San Francisco, has made a more specific point. If a genuinely dangerous pathogen ever does re-emerge from thawing permafrost, the far more likely path isn't a single infected mine worker triggering a global outbreak. It's a dangerous pathogen re-entering wild or domestic animal populations, first quietly, long before anyone in a city notices anything at all. That's not a reason to stop paying attention. It's a reason to be worried about the right thing. This is the question that keeps biosecurity researchers up at night. Could a revived, tens of thousands of years old zombie virus trigger the next human pandemic? The scientific consensus is more reassuring than the headlines suggest. Almost certainly not, at least not this. Every giant virus pulled from the Siberian permafrost so far is genetically tuned to hunt single-celled amoebas. Human immune systems, body temperature, and cell structure make our tissue functionally invisible to them. That's not a guess, it's the reason Clavery's team has felt comfortable reviving them in a lab in the first place, the same reason they picked a harmless amoeba as bait, rather than risk anything closer to us. The real risk isn't the giants at all. It's what else is buried beside them. Scattered across the Siberian tundra are the preserved remains of ancient hominids, prehistoric hunters, and extinct animals, and inside them, uncharacterized classical pathogens that we know evolved to infect mammals, not amoebas. If industrial mining breaches a burial site like that, dormant strains of ancient smallpox, influenza, or unknown zoonotic bacteria could become airborne and reach human workers before anyone even knows they're there. Siberian permafrost isn't just frozen mud. It's the planet's own cryo-archive, a fragile 50,000-year balance of deep-time biology that has stayed sealed for reasons that had nothing to do with us. So here's where that leaves us. Giant viruses can survive at least 500 years frozen and reactivate on contact with a living host. That's proven repeatedly in a controlled lab. None of the ones we've found so far can infect a human being. The thing actually digging into that ancient ground fast enough to matter. Isn't the climate its industry, moving at a pace geology never had to plan for? And the real unknown isn't the amoeba killers we already understand. It's the pathogens we haven't found yet, sealed inside the things and the people buried next to them. The greatest threat to modern civilization was never going to come from some far-off, futuristic source, a lab-made bioweapon, a rogue AI. It may come from something much older and much closer, our own machinery, carelessly breaking into an Earth that existed long before humans built a single city or wrote a single word. It took humanity thousands of years to learn how to build the world above ground. Our next test might be learning when to leave the world below it exactly where it is.